The overall goal of this confocal microscopy-based ratiometric imaging technique is to determine and visualize extracellular pH in bacterial biofilms in real time.
This method helps to answer some key questions in the field of biofilm research, such as where exactly acid production in biofilms occurs, and how pH microenvironments are conserved over time in distinct areas of the biofilm. The main advantage of this technique is that extracellular pH can be recorded in real time with high spatial resolution in all 3 dimensions.
To carry out confocal microscopic calibration of the ratiometric dye, use an inverted confocal microscope equipped with an incubator, a 63x 1.2 numeric aperture water immersion objective, a 543 nanometer laser line, and a metadetector. Prepare Heaps buffer stock solutions adjusted to pH 4.5 to 8.5 in steps of 0.1 pH units. Then pipette 100 microliters of each solution into the wells of a clear bottom 96 well plate for fluorescent microscopy. While wearing nitrile gloves, add 5 microliters of a 1 millimolar stock solution of C-SNARF4 in dimethyl sulfoxide to each well of Heaps buffer. Then place the 96 well plate on the microscope. Next, turn on the microscope and open the microscope software. Click on the following panels: Acquire Laser, Acquire Micro, Acquire Config, Acquire Scan,